Hello everybody, my name is Amjad and I lead the development of bioinformatics capabilities here at Scilligence. Today I'm excited to present to you on Molbio, a solution that we're developing in-house to address molecular biology needs. Today's agenda is as follows. We'll begin with a brief introduction on what molecular biology is. After that, we'll dive right into Molbio, looking at some of the current capabilities that the software contains right now. And then we'll end with a short slide on what we hope to implement in the software going forward. All right, so a quick primer on what molecular biology is. Well, this is the branch of biology that deals with the structure and function of macromolecules, primarily genes and proteins. And as such, most molecular biologists focus on genes and proteins. Why? Well, because genes contain the information that is required to make proteins, nucleic acids like A, T, C, and G. And proteins are the effector molecules within the cell that will perform key functions. Now, both genes and proteins are sequence-based entities, and as such, information is contained at the sequence level. So knowing this, we've developed Molbio with a few things in mind: easy, fast, and smart. What we mean by that is we've adopted a design philosophy to make the UI as intuitive as possible so that end users do not need much training and can get onboarded quickly. We want the software to be fast, so it's responsive and it saves you time with your complex workflows. And of course, we want the software to be smart. What we mean by that over here is that there is biointelligence built into the software, so it's able to notify you and alert you based on the workflows and based on different errors that you may encounter. At the same time, Molbio is also being built from the ground up in a way that it allows us to scale and deliver capabilities for advanced molecular biology workflows. The field of molecular biology has come a long way in the last 50, 60 years, and we hope to be able to accommodate these complex workflows in the future. In this slide, we're depicting a typical molecular biology workflow. Please note that labs might do things differently. This is to show you a representative molecular biology workflow that may be commonly employed in most molecular biology labs. We begin with the blast search. You can imagine we have a sequence of interest that you want to search across databases to see if it has um, significant hits in other species, homologs, so to speak. A second parallel step may be sequence alignment, where you have a more refined search of this, and now you're looking or comparing the sequence of interest across more closely related sequences. Of course, once you have the sequence of interest identified and confirmed, you might want to move on to the cloning workflow, which is where you prep your insert for cloning into a vector, the ligation processes where they come together. We often make use of bacteria for transformation. They are able to uptake this foreign plasmid. They can be grown selectively in culture media. And then, of course, sequencing, or the very last step, allows us to identify which of these bacteria harbor our vector or plasmid of interest. Now, you'll notice on the bottom we have three boxes: sequence display, sequence manipulation, and biology calculators. These are our broad classifications of functions that must be contained in Molbio for it to be a successful software for use with the common molecular biology workflow. And so we are depicting or representing at each of these steps, at each of these stages, the various steps that may involve one or two of these functions. So what can Molbio do right now? Some of the things that the software is currently capable of doing are listed over here. Sequence display, the idea being that the end user can deposit a sequence, and of course play around with the various visual tools at your disposal to see how best they want to depict and represent that sequence on screen. Sequence manipulation, we can not only just create sequences but also edit them at any time. We connect to AddGene's knowledge base database to auto annotate features, but should you choose, you can always add your own custom features and primers and enzymes. We have capabilities of BLAST and sequence alignment, both of which we will show in today's demo. BLAST, of course, for checking homology across global and local databases and then finding regions of local similarity. Sequence alignment being somewhat similar, but of course focusing more on specified sequences. We also have the capability to handle basic learning workflows, and we will be able to show that today as well. And of course, as all of our other apps, um, giving admins and giving companies, the ability to manage projects and users and thereby implement security and access. In this section, we'll give you a brief rundown of the sequence alignment capabilities. This is, of course, a work in progress, so please note that. We're going to select Run Alignment, and we'll be taken to this web page where we can enter information on the job. In this case, I am going to be aligning sequences for a protein called SUMO. I can add sequences through two primary methods. The first one is shown over here, where if I have previously deposited or registered entities, in this case SUMO1 from human and SUMO1 protein from mouse, I can pull them from Molbio. They're auto-suggested, and I can simply add them to the tray. A second method is to directly copy and paste sequences into the tray, as you've seen over here. The file format that is currently accepted has to be FASTA. So you can see them, the various model systems from Arabidopsis to zebrafish to Xenopus are listed over here. We are going to select the algorithm as clustal, and we can also switch the sequence type to protein because these are, of course, amino acids. On the bottom section, you will see that the end user has the ability to modify a few of the various run parameters. Right now, we support clustal and MAFT. We hope to add additional algorithms in the future along with additional parameters for run as well. I'm going to select %ID for today's demonstration and select the file type as clustal and then click on Run Alignment. When I do this, you will notice this progress box that appears that tells us where we are in the process, and it's a very fast process. So once it's done, we can click on Open Alignment Results, and it takes us to this separate page where you can view the results. You also have the ability to save these results, so you can come back to them, and of course download them in a different style if you like. We begin with the BLAST function. BLAST stands for Basic Local Alignment Search Tool. It allows us to take our sequence of interest, a protein sequence in this case, and align it against other sequences to check for homology. There are two different ways we can paste our sequence into the tray. The first one is simply to paste it into the sequence tray that you see in the background. Just a Control-C, Control-V, copy and paste will be sufficient. For today's demo, what I'm going to show you is use the NCBI ID. So if I enter the ID over here and click Search and Add to Tray button, we're going to connect to NCBI or UniProt via an API key, and in just a moment we can pull in the sequence from these websites. So you can see it populate in the sequence tray. The first line, this is in FASTA format. The first line is indicated by a triangle bracket containing the metadata, and then the second line has the actual amino acid sequence for my protein of interest. We can select various target databases at NCBI, but for today's demo, we're going to be checking it against locally deployed sequences that I've just used my clipboard to copy and paste. I'm going to give the job name. I'm going to give the task a job name, so it's going to be the same protein name with just the word test after it. Bear in mind this is a protein sequence, so sequence type is protein, and of course for that reason we'll be using the BLAST-P program. The E threshold is going to be set to 1. This is simply a statistical significance that lets us know how significant a particular match is to our query sequence. There are 4 different output methods. I'm going to choose a text file. It's just easy to read and certainly easier to append to an experiment once I'm done. I'll click the Run BLAST. It takes a few moments based on the number of sequences deposited. But you see the results are produced over here in this text format. We have the references listed above, and then as we slowly scroll down, you can look at our query sequence that was used along with any other sequences producing significant alignments. And as you can see in the very right hand column, the low E numbers indicate high significance. As I scroll down, you can see matching between our query sequence versus all of those indicodant sequences that were used in the local database. And we are done with this functionality over here. The next step will be to do the sequence alignment. Of course, Molbio is not going to be static software. What we'd like to be able to do in the future as we keep working on this software is to add in more useful features. One of them, for example, is the relationships. Currently, in our registry module, RegMol, we are able to capture a visual history of entities. We'd like to expand upon that and record even more of a visual audit trail that shows different pieces of DNA coming together to form a particular plasmid and how all of that is linked together along with all the relevant metadata. We're continuously adding more cloning with methods. For now, we support GC, TA, and the traditional restriction digestion cloning. But you can see as advances in molecular biology are made, we'd like to be able to support advanced methods such as Gibson assembly or perhaps the at-R methods developed by Invitrogen for their gateway cloning. Primer design, of course, right? So we live in a world, of course, where everybody's, well, most people, I should say, are familiar with CRISPR-Cas9. A lot of these kind of experiments require analysis of the sequence to be able to design primers that are useful for the workflow. So how do we calculate these properties? How do we, what kind of calculations are made? How do we analyze the sequences? We have some of this already contained within Molbio, but even more biointelligence over here would certainly be helpful. Coming back to our molecular biology workflow slide, what I'm showing right now is a third panel on the very bottom that indicates what kind of apps we anticipate working with each other at each of these steps. And of course, this is not an exhaustive list. This is simply a quick summary of what we think right now might be the case in our current platform. So for example, for BLAST, sequence alignment, vector prep, and the insert prep, we expect Molbio to work seamlessly with the ELN, where you as a molecular biologist can capture information at each of these steps within your electronic lab notebook as well as to how a particular sequence of interest was identified, aligned, and how preparations for cloning were made. Once we are at the stage of transforming cells and we have cell lines that have been successfully transformed, we can push this information to our registry software, RegMol. We can even capture information on these cell lines, these bacterial colonies in inventory. And then similarly, the same case for sequences where we have assay results being able to push back to RegMol as well. So this is not an exhaustive list again. This is simply a brief showcase of what existing apps we expect to work with Molbio. We will continue building upon this, expanding on this, and you can certainly see additional modules come into play over here to give you this complete workflow from end to end. Of course, no presentation can ever truly end without giving the requisite credit for its due. I'd like to thank the lead developer for Molbio, Dongchang. He's been working closely with me for the last year and a half working on Molbio. Truly phenomenal work. I really appreciate him putting so much effort into this. We've had some of the newer dev team members join us for BLAST, Ray, for sequence alignment, Tim Greiser. I'll also take some time to thank Alan from the sales side because he's given me quite a lot of client feedback on what clients are clamoring for right now, what our leads want, and this is certainly helpful in informing us when it comes to developing the software. And then for the development feedback itself as to how we go about developing the software, I'll certainly extend my gratitude to Tommy. With that, I'll also thank you, the audience, for your time and attention, and we will be happy to take any questions you may have.